The Production of Butanol by E. coli and its use as a biofuel, created by Alexis Williams, Matthew Shook, and Kathleen Davis. A biofuel is a fuel derived from biomass or biological waste. Some strains of bacteria and yeast can use starch to produce a type of biofuel called butanol through fermentation. Louis Pasteur first recorded butanol production via anaerobic bacterial fermentation in 1861. Around 1910, there was a shortage of natural rubber. This created interest in butanol production as it is used to make synthetic rubber via isoprene as an intermediate. Between 1912 and 1914, Chaim Weizmann isolated a strain of bacteria, Clostridium acetobutylicum, which yielded more butanol and acetone than previous strains of bacteria. Weizmann then patented the first ABE process in 1915 using Clostridium acetobutylicum, maize, and potato. ABE fermentation is the microbial fermentation of starch to produce acetone, butanol, and ethanol. Acetone was in high demand during World War I because of its use in ammunition production. Therefore, the US and UK put Weizmann in charge of mass-producing acetone using his ABE process. This push immediately put acetone, butanol, and ethanol at industrial production levels. After the war, Weizmann's patent expired and ABE plants opened in many parts of the world, and were used to produce many commodities like synthetic rubber and car lacquers. In the 1960s, the petrochemical industry was emerging as the more cost-effective solution, and the ABE processing plants soon shut down. In recent years, concern about environmental damage from burning fossil fuels, the push for renewable energy sources, and the rising cost of gasoline have all helped increase the interest in biofuels, including butanol.
Since Clostridium acetobutylicum had been used historically to produce butanol via ABE production, scientists began studying the metabolism of C. acetobutylicum with the goal of figuring out how to increase its ability to produce butanol. In C. acetobutylicum, like most organisms, glucose is converted into pyruvate through glycolysis, the first step in the metabolic process through which the cell breaks down carbohydrates to produce energy. The pyruvate produced during glycolysis is then converted into acetyl-CoA through a process known as decarboxylation. In C. acetobutylicum, the acetyl-CoA produced during decarboxylation is then converted into acetyl-acetyl-CoA by an enzyme called acetyl-CoA acetyltransferase, abbreviated THL. Acetyl-acetyl-CoA then goes through a 7-step process through which it is converted into N-butanol. Scientists weren't able to promote significant increases in C. acetobutylicum's ability to produce butanol, so some scientists took the genes from C. acetobutylicum that coded for the enzymes in butanol production and inserted them into E. coli cells, so that the enzymes would be expressed in E. coli. Just like in C. acetobutylicum, glucose is converted into pyruvate through glycolysis, and pyruvate is converted into acetyl-CoA through decarboxylation. So when scientists inserted the genes for THL and the other fermentation enzymes into E. coli cells, the E. coli was able to produce butanol through fermentation. So, how exactly did scientists insert genes from C. acetobutylicum into E. coli? Scientists can insert DNA from one type of bacteria into another through a series of steps collectively known as recombinant DNA technology. The first step in recombinant DNA technology is the removal of a gene or genes of interest from a particular organism. In this case, the C. acetobutylicum genes that code for the enzymes involved in fermentation were removed using restriction enzymes such as EcoR1. A circular piece of DNA is called a plasmid, and it includes a gene for antibiotic resistance. It is also cut with the same restriction enzyme. The cut plasmid and the removed genes are hybridized with an enzyme called DNA ligase, which joins the cut DNA ends together to form a new circular piece of DNA known as recombinant DNA. The recombinant DNA is inserted into E. coli cells through a process called transformation. After transformation, the E. coli cells which have successfully taken up the plasmid have both the plasmid and their own chromosome. The transformed cells are plated onto a medium containing the antibiotic, so that only the cells that have successfully taken up the plasmid, which has the antibiotic resistance gene, can grow on the medium. To get the recombinant E. coli cells to produce butanol in the lab, they are grown in bulk. Liquid media is inoculated with the recombinant E. coli cells and grown in a shaking incubator at 37 degrees Celsius. After the cell cultures have incubated for a while, IPTG is added to the liquid media containing the growing cells. IPTG is a compound that mimics allolactose, and so it can trigger the expression of the lac operon or any genes in front of it. The recombinant DNA plasmid in the E. coli contains the lac operon, so when IPTG is added to the media, it stimulates the expression of the genes that code for the fermentation enzymes. After the fermentation has proceeded for at least 8 hours, the cell cultures can be removed from the incubator and the amount of butanol produced can be measured. To measure the production of butanol by the E. coli, a small sample is taken from the growth culture and placed in a centrifuge, where it is spun at high speed so the E. coli cells settle to the bottom of the tube and form a pellet. The liquid above the pellet is called the supernatant, and it contains the butanol, which has been released by the E. coli cells. A small volume of the supernatant is then taken and analyzed on a gas chromatograph, like this one, and the butanol in the supernatant is quantified by flame ionization detection. When scientists did this, they obtained a yield of 0.5 grams of butanol per liter of E. coli cells. This is not a very high yield, so scientists tried different ways of increasing the yield. The most success was had by Dr. Chang and her team of scientists at the University of California in Berkeley. As it turns out, when all 5 enzymes from C-acetobutylicum were put into E. coli, much of the butanol being made during fermentation was being broken down into chemical precursors through reverse reactions by the fermentation enzymes. Dr. Chang's lab solved this problem by substituting some of the fermentation enzymes from E. coli with similar fermentation enzymes from other organisms. So instead of putting all 5 fermentation enzymes from C-acetobutylicum into E. coli, they took 3 of the enzymes from C-acetobutylicum, 2 enzymes from Ralstonia eutrophis, 1 enzyme from Arenomus caviae, and 1 enzyme from Treptonema denticola, and inserted these 7 enzymes into E. coli to create a hybrid fermentation pathway. The enzymes in this hybrid pathway have reverse reactions that are so slow that very little of the butanol product is lost through conversion to its chemical precursors. Dr. Chang's lab was able to obtain a yield of about 5 grams of butanol per liter of E. coli, and Dr. Chang is confident that additional modifications can be made to increase the yield of butanol to somewhere in the range of 10 to 15 grams per liter of E. coli, which would be a high enough yield to design a modern industrial process around. Hypothetically, what would such an industrial process look like? Well, E. coli is already used to produce ethanol, another type of biofuel, via fermentation on an industrial scale. This process involves incubating the E. coli with feedstocks such as corn grain, sugar beets, or sugarcane in large containers with no oxygen called fermenters, and then separating the produced ethanol from the E. coli cells, feedstock material, and other byproducts, including water. Theoretically, an industrial process for producing butanol from E. coli via fermentation would be similar. The same type of feedstocks can be used, and like for ethanol production, butanol production would also have to take place in anaerobic fermenters. Like in ethanol production, the butanol product would have to be removed from the mixture of E. coli cells, feedstock material, and other byproducts. This removal and purification process would be similar to that for ethanol, but slightly different based on the chemical differences between butanol and ethanol.
An important advantage of butanol as a biofuel is that in many cases it can be used to directly replace gasoline. As you may be aware of, some cars today run on flex fuel or E85, which means it uses a mixture of ethanol and traditional gas. However, ethanol doesn't contain as much energy as gas per volume and cannot be used at high concentrations or it could cause traditional engines to fail. Butanol has similar energy density to gasoline and can be used in current combustion engines without modification.
The production of N-butanol in E. coli offers many economic and environmental advantages and should be a major strategy toward a renewable source of energy for the future. However, it will be necessary to improve the process of production to make it easier, more efficient, and less expensive. There is also a need to increase the amount of N-butanol produced by the process in order to meet our diverse energy needs, such as jet fuel, diesel, gasoline, and industrial chemicals. Fortunately, scientists have already made advances in this area through several research strategies. There are three primary strategies that are the focus of research to increase and improve the production of butanol by E. coli bacteria. They are metabolic engineering, gene transfer techniques, and protoplast fusion to develop hybrid bacteria. As described previously, the glucose metabolism of a bacterium such as E. coli can be altered by cloning a gene from Clostridium or another bacteria and using plasmids to express it in E. coli. This way, the enzymes that these genes code for are produced in E. coli, and it is able to make butanol through this new synthetically engineered metabolic pathway. In addition, E. coli possesses an alternative pathway that results in N-butanol production. Gene transfer techniques have been used to modify this pathway to increase the production of upstream precursors, ketobutyrate and ketovalerate for the ketoacid pathway, so that more N-butanol can be produced. A second major strategy has been to improve the resistance of E. coli to N-butanol. Currently, once the biofuel is produced, it can be toxic and damaging to the bacterial cell membrane. What is needed is to improve the resistance to the bacteria. Scientists have used gene transfer techniques that modify E. coli and have proven that it can withstand higher concentrations of butanol and continue its production. Finally, another technique used to improve the yield has been to develop hybrid bacteria that can produce more butanol and tolerate even higher concentrations. Scientists had observed that lactobacillus bacteria were resistant to the toxic effects of butanol, but that the bacteria lacked enzyme systems necessary to produce the biofuel. Using protoplast fusion techniques, a hybrid bacterium has been created that has the characteristics of E. coli in producing butanol and the resistance to the toxicity that lactobacillus demonstrates. This graph shows that the hybrids tolerate high butanol concentrations. Currently, the genomic shuffling strategies are under investigation to improve the production of butanol by this hybrid even more. These various strategies using E. coli to produce butanol will clearly improve the amount and efficiency of butanol production. However, there are challenges to butanol production that must be addressed. These include low product yield, stability issues, expense, and unwanted byproducts. Possible solutions to the dilemma include the use of lignocellulosic hydrosylates, or non-food resources, knockout genes, improved factory hygiene, and gas stripping, pervaporation, or extraction techniques. Fortunately, we have many other possible sources for biofuel production to meet the energy needs of the future. Not only can we utilize several bacteria in production, but the ways that bacteria can be manipulated to result in energy sources are also numerous. The future for renewable energy sources is bright.